During the summer, male Pacific walrus utilize terrestrial haulouts to rest between foraging bouts in Bristol Bay, Alaska. Currently, there are about 5 known haulouts in this region, but why animals move between haulouts and forage in this area is unknown. Researchers from the Alaska SeaLife Center partner with Point Defiance Zoo and Aquarium to dive in and learn more.
We really wanted to know if forage availability was what was changing the movement patterns or if that plays a role. So in Bristol Bay, there's also an increase in human presence, increase in commercial fisheries, and also there's potential interactions from those developments. And so there's other drivers that might be happening as well, but it's really challenging to explore forage availability as one of those drivers.
So walrus are foraging on sessile animals that don't move, primarily bivalves or crustaceans or things that are in the benthic sediments. So you can imagine as a walrus kind of moves through the environment, they're hoovering up all of the shells in an area, and at some point there won't be any prey available in that localized area anymore to support them.
To understand how much prey is available, our team needs to gather an idea of what lies on the ocean floor.
The primary method that's been used to study benthic ecology, so ecology of things at the bottom of the sea, is really bottom grabs or core sampling. So bottom grabs, the most common one is called a Van Veen grab, and if you kind of imagine kind of a bulldoze or shovel opening and closing like that, they're dropped off of a research vessel, they take a big scoop of the sediment, you bring it up, and you basically have trays and trays of muck and clams and crabs and polychaete worms and all this stuff that very skilled identification aquarists or research scientists can go through and ID out and tell you what was in that small sample.
This is really logistically challenging because you're fighting with weather for ships to go out, the logistical challenges of deploying large pieces of equipment, and it's really costly. Obviously, as you can imagine, in Alaska, these prices add up really quick. So is there a way that we could use some more of a novel or modern technique to get at the same answer of presence, absence of species at the benthic community? And could we maybe get at somehow assessing their quantity as well?
To mitigate some of the obstacles associated with traditional methods, our researchers turned to a different approach to study prey availability by using environmental DNA.
eDNA is short for environmental DNA, and basically it's DNA that comes off of any living being as part of their normal life process. So if you think about almost like a crime scene, everything that we touch during a day has some of our cells wash off on it. If we're sneezing, if we're spitting, if we're urinating or defecating, all of that has our DNA in it, and it enters our known environment. So for animals, it's the same thing. All of their cells and their bodily processes shed DNA into their environment. And so it actually gives us an opportunity to try to determine if animals have been in an area, what species of animals are in an area, timing, things like that, without actually having to spend a lot of money to go out and sample them, collect them. A lot of it can be done just by sampling a cup of water.
With these curiosities in mind, our team sets out for Kitsitna Bay, Alaska. Although outside of known walrus territory, Kitsitna Bay is still an ideal spot to test the validity of our eDNA approach for detecting bivalves. While our team prepares to dive in, let's hear more about the sampling process from one of our divers.
So when we were diving in Kitsitna Bay, we were taking water samples from the area that we were then going to take a sediment sample with the hope of finding bivalves within that sample. 20 to 30 feet was our typical depth, and we're looking for a flattish bottom profile with appropriate substrate where we hope to find bivalves. The main hurdle that we had to overcome was the fine sediment that we were dealing with, both combination of glacial silt and mud, that when we dove down, as soon as we disturbed that substrate, it basically created an impenetrable cloud of sediment that we couldn't see through and really couldn't work in very effectively. So we made the decision to, instead of trying to sort and find bivalves underwater, to just collect a volume of sediment in a given area and then sort that up on the deck of the boat where that visibility then wouldn't be an issue.
Our sample sites are teeming with life, so how can we sort through all the eDNA to target walrus prey items like bivalves? Join us next time as we take our field samples to the lab for a closer look at unraveling eDNA.